Hello students. In this session, we are going to solve AIET 5 questions which we find that you might have found difficult during your examination. So let us start with our first question. So our first question is question number 102. When a patient with a defective ADA was treated, okay, which of the following steps were performed for the gene therapy? And our statements are lymphocytes were obtained from the patients, lymphocytes are transferred to the culture tissues, lymphocytes were transferred with a normal ADA gene, the transfected cells were are returned to the patients. So all of these are the first option, and then our second option is only C and D, and our third option is only D, and then our fourth option is kid cannot be treated. So let us look at this question. The question says when the patient is detected by the ADA or when they have ADA deficiency. ADA is, that's adenosine deaminase deficiency. So here what happens is a person lacks this adenosine deaminase, an enzyme, which is responsible for the immune function. So in the absence of this enzyme, our immune function fails. That leads to the failure of our immune system to detect any of our antigen cells, that is external agents. So when this happens, our body fails to differentiate between uh, any of the antigens, right? So that will lead to the failure in understanding if we have a disease or if anything is infecting us. So that will lead to the failure of immune system, which leads to a condition called as SCID, that is a severe combined immunodeficiency. It's a syndrome. SCID is a syndrome because it shows multi-symptomatic characteristics. So now, according to the treatment for the ADA, we know that the gene therapy is employed for the ADA treatment. So where we collect the lymphocytes from the patients, and then we alter their lymphocytes by using we culture them in the dishes and then alter these same lymphocytes with ADA gene, and then it is transferred to the patients. Correct, right? So all of these steps are very uniform and it happens one after other. So all of the above are correct. So that is why I go for all of these. Now coming to the last option, that is SCID cannot be treated. We have treatment for SCID. Apart from the gene therapy in SCID, that is severe combined immunodeficiency, we go for enzyme therapy, or in some cases we go for the bone marrow transplantation. Okay, so that is why our option D is wrong. Okay, coming to the next question. Read the following statements. So we have our following statements. Mango trees cannot grow in temperate countries like Canada and Germany. Snow leopards are not found in Kerala. Tuna fish are rarely caught beyond tropical latitude in the ocean. Average temperature exceeds 100 degrees Celsius in the thermal springs and hydrothermal vents. In polar areas and high altitudes, temperature goes up to 70 degrees Celsius. Temperature goes to greater than 50 degrees Celsius in tropical desert in summer. Okay, which of the following above statement is false? And we have our options only A, and B and C, only E, and then we have E and F. So these are our options. So when we look at this question, let us look at the first option. Mango trees cannot grow in temperate countries like Canada and Germany. So this statement is correct because we know that mango is for tropical countries. Okay. Snow leopards are not found in Kerala. Yes, they are found in Himalayas, not in Kerala. Tuna fish are rarely caught beyond tropical latitude in the ocean. Correct. They are mostly caught within the tropical latitudes than beyond tropical latitudes. Next one, average temperature exceeds 100 degrees Celsius in the thermal springs and the hydrothermal vents. So this is also right. You know that, right? In hydrothermal vents, the temperature can go up to very high temperatures. Next, in polar areas and high altitudes, the temperature go up to 70 degrees Celsius. Now it's given plus 70 degrees Celsius. In high altitudes, it goes minus 30 to minus 50 degrees Celsius. So this option is wrong. Temperature goes to greater than 50 degrees Celsius in tropical desert in summer. So this option is also right. So our option, that is E, only E, we have it in option C. So our option C is correct. Let's go for the next question. Which of the following is genus of extinct vascular land plants of Devonian period and is thought to be a part of the group from which the modern ferns and the seeded plants evolved? So our options are Zoosterophyllum, Rhynia, Xylophytum, and then we have lycopods. When we look at this question, we have to find the genus of extinct vascular land plants. So extinct vascular land plant is Psilophytum. Okay, so this is known to be the ancestor of all the ferns and the seeded plants that have evolved. Okay, they are found in Devonian period. When we look at Zoosterophyllum and then we have Rhinia, both of them were found in Devonian period, but they are not the ancestors of what we see the current vascular plants. Now, lycopods represent ferns. Okay, so they are not related to this. All are vascular plants only, but the current ancestor of the ferns and the seeded plants that what we have evolved is through Psilophytum. Okay, so our option is option C. Next question. If for some reason the vasa efferentia in the human reproductive system gets blocked, gametes will not be transported from. So our options are epididymis to vas deferens, okay, seminiferous tubules to vasa efferentia, then we have ejaculatory duct to epididymis and testis to epididymis. So let us look at this question. What happens if for some reason vasa efferentia in the human reproductive system gets blocked? So if you look at the cross section of the testis, we know that within the testicular lobules we have seminiferous tubules. The seminiferous tubule combine together to open up into a larger duct eventually. Okay, so these larger ducts, around 8 to 12 ducts are there, and these ducts are called as vasa efferentia. Vasa efferentia. Efferentia means inside to outside. So this vasa efferentia that is carrying the sperms will eventually lead to the outermost surface of the testis through epididymis. Okay, so this is epididymis. So this is epididymis and this is vasa efferentia. Okay. Now, if this gets blocked, what happens? The sperms will not come from the testis to the outside. So that is why our option is testis to the epididymis. From the inside of the testis to come out of the epididymis, you need vasa efferentia. If vasa efferentia is blocked from the inside to outside, the sperms will not come out. Okay. So that is why our right option for this question is option D, that is testis to epididymis. Understood? So our next question is we have to match column 1 with column 2 and identify the correct option. So let us look at column A and column B. In column A, we have primers, separation and purification of products, okay, and then we have precipitation of DNA and transformation. In column B, we have PCR, ethanol, uptake of the foreign DNA by bacterium, and downstream processing. And the options for these questions are given here. Okay, hold on, sorry. That is A, B, C, D. So let's take a look at it. Okay, so let's go and solve this question. Column A, primers. What are primers? Primers are short complementary sequences of your DNA. We know that our DNA has 2 strands, correct? Now let us say that we need to create a daughter strand. To create daughter strand, we need DNA polymerase, correct, right? Which is an enzyme that helps in creating new strand of DNA. So this DNA polymerase cannot synthesize the DNA on its own. Why? Because it requires a free 3' OH. Free 3' OH means it requires the end, 5' and 3', right? So it requires this 3' end. So how do we get that 3' end? Through these short primers. What are these primers? If my strand AACCG, if this is my strand, one parent strand I have 5' to 3', then what will be my primer? The primer will be ATTGG. Okay, let us take that this TTGG is a short sequence. So the short sequence is complementary to the parental DNA. So these are called as primers. Where do we use primers? We use primers in polymerase chain reaction. When does it happen? We know that after denaturation, during annealing process, we add artificial primers, and these artificial primers in the extension process, they will be used by your DNA polymerase to create a new daughter strand. So that is PCR reaction. So I'll write it as A. Next one, separation and purification of products. So in biotechnology, after we have created any biological products for human use or medical or any use, we know that the purification of the products have to be done. So this purification and concentrating these products comes under a special, a special field which we call it as downstream processing. Okay, so that is why we put it as B. Okay, I'll write it cleanly. This is B. Okay, next one, precipitation of DNA. So while if you have to isolate a DNA, we know that DNA is within the cell, and we go for enzymatic digestion where we get rid of all the cellular constituents in the form of biomolecular digestion, and eventually it leads us with only DNA. But DNA is completely, it looks dissolved within this digestive enzymes. So now what we do is we start adding chilled ethanol. When we add chilled ethanol, the ethanol goes and binds to this DNA strand and removes water. So dehydration happens. When the DNA gets dehydrated, it starts precipitating, and while spooling, we can isolate. So we use ethanol for precipitation of DNA. I'll write it as C. Next one, transformation is any of your bacterial friends. If they have taken up the foreign gene, they are called as transformed cells or transformed bacterium. So that is why I'll write as B. Okay. So let's see which is our right option. That is A is 1, B is 4, correct? Yeah, B is 4. Here we have. Okay, yeah, C is 2. Yeah, C is 2. Okay, our option is D. Okay, D is the correct option for this question. So we have another match the following question. So in this question, we have column 1 and column 2. So let's look at column 1. Column 1 says sacred groves, cryopreservation, biodiversity hotspots, and endemism. Column 2 says germplasm of threatened species can reduce mass extinctions, species confined to a particular area, last refuge of rare plants. So when it comes to sacred groves, we know that they are the very important plants that are preserved in a particular area, right? So that is why sacred groves are the last refuge for all rare plants. So I'll let it as A. Cryopreservation. It is a scientific technique where we preserve this organic material such as tissues, cells, or any biological products at a very, very low temperature, such as minus 190 minus 190 degrees Celsius, using liquid nitrogen. Okay, so this process is called as cryopreservation. Here, for saving this genotype of a particular organism for several generations, okay, so instead of storing the entire species, what we'll do is we can take small tissues such as germplasms and preserve it under very low temperature for a very long period of time. So that technique is called as cryopreservation. So I'll write it as B. Biodiversity hotspots. Biodiversity example is Western Ghats in our India, which is present in South India. So if you look at this, we have a very huge species diversity and we have a very large ecosystem. Okay, so that usually can be a hope for preventing any of the mass extinction or sudden loss of biodiversity. Okay, so that is C. So can reduce mass extinction. Next one is endemism. Endemic species or endemism are the organisms which are restricted to a particular habitat. For example, you have snow leopards in Himalayas, right? And then these are called as endemic species. Okay, kangaroos from Australia. So this is D. So let's look at our options. Okay, A, B, C, D. So we know that our A is S, sacred groves, last refuge for plants, and cryopreservation is preservation. That is B is P, C is Q, and then D is R. Okay, so our option is A for this question, 120. Okay, so let's look at the next question. And our next question is, which of the following is most likely reason for the break in menstrual cycle in females? So we have our option: fusion of pronucleus, maintenance of the hypertropical endometrial lining. Okay, then we have maintenance of high concentration of sex hormones in the bloodstream. Next one is retention of well-developed corpus luteum. So when does the menstrual cycle break in females? So we know the process of fertilization and implantation, but let us take a look at it. If you see, what happens is the male pronucleus gets used by the female pronucleus to finally fertilize and create a zygote. Now, once the single cell zygote starts dividing into 2, 2 to 4, 4 to 8, okay, which finally becomes morula, and it flows through the fallopian tube during this journey and enters the uterus. Here, what happens is our morula is developed into a blastula, where it is differentiated into trophoblast, and then we have innermost cells, trophoblast layer and innermost cells. So this trophoblast layer have two layers, that is cytotrophoblast and syncytotrophoblast. So the one of the trophoblast layer will try to go and attach to the walls of uterus, and the other trophoblast layers, what they will do is they will produce a human chorionic gonadotrophin, okay, HCG, and this HCG will, what it does is it will prevent our corpus luteum from degenerating. So when our corpus luteum do not degenerate, now what will happen? That will lead to the production of more progesterone. Okay. Okay. So it will produce more progesterone. When progesterone is more, now what will happen? The maintenance of the endometrium lining will take place. So when the endometrium lining maintenance is taking place, and then you have your corpus luteum continuously producing progesterone, now what else happens? Now other hormones are also released, okay, which help in the proliferation of the cells of the blastula. So here, to stop the menstrual cycle, what should happen? Fusion of pronucleus has to take place. Once the fusion of pronucleus has taken place, so that will lead to the following events. Okay, next one. Which of the following is correctly matched? So we have alpha diversity, number of species in a given habitat, and then genetic diversity, variation of genes within species. Then we have beta diversity, diversity of habitat in the whole region. Next we have is species diversity, the product of species richness and evenness. So let's look at the first option. We have to find the incorrectly matched pair. Alpha diversity. When we talk about alpha diversity, they say that number of species in a given habitat. This is right. Alpha diversity refers to the species diversity or species richness in a given area. Now, genetic diversity, variation of genes within the species. For example, we have a species, and if we have many variants within them, that is called as genetic diversity. Okay, that is also right. Beta diversity, diversity of habitat in the whole region. Now, when we talk about beta diversity, just imagine one thing, okay? Now, we have a community, a group, okay? Now, this group, how fast it can be replaced by another group, okay? Let us say we have a community A and community B. Now, how fast community B can be replaced by community A, or community A can be replaced by community B? So, whenever this complete community can be replaced by others, so that is called as beta diversity. Where can you find beta diversity? Where there is lot of community richness or species richness. You have lots and lots of species. Now what will happen along the direction? Now what will happen? These small communities or whatever the communities get replaced by others. So this is called as beta diversity. So that is why this is a wrong option. Whereas species diversity, yes, the product of species richness and evenness, how much of species are distributed and how evenly they have spread, that is called as species diversity. So our option is C for this question. Let's go for the next question. Given below are three statements, each of one or two blanks. Select the option which correctly fills the blanks in any two statements. Our first statement says, diseases or infections which are transmitted through sexual intercourse are collectively called as dash diseases. Genital herpes or genital herpes is dash disease. Sterilization in male is dash while female is dash. I know that the options are very easy. The question is very easy, but the options are quite tricky in this question. See, we have to find the correct match according to our question. So let us look at the first option. Diseases or infections which are transmitted through sexual intercourse are collectively called as what? Venereal diseases. Correct, right? They're called as venereal diseases. Next one. Genetic herpes is incurable. Okay, genital herpes is actually incurable disease. Okay. Next one. Sterilization in male is called as what? So a vast difference will be cut and tied. That's why it is called as vasectomy. whereas you have fallopian tube being cut in the female, that is why it is called as tubectomy. Where do we have the third option as vasectomy? Both the options have tubectomy, so we reject both the options. Okay. Whereas here it is curable is given. So since it is not curable, our option A is correct. Okay, we don't have the right options for the third statement. So that is why we eliminate the third statement and we go for option A, that is venereal and incurable. Next question. We have a lot of statements to read. Question number 129. Sperms cannot fertilize an egg unless they have undergone to capacitization reaction in the female reproductive tract for several hours. In vitro studies indicate that the sperms can fertilize the ovum if they are first washed before being introduced to the ovum. Now, these observations suggest that the process of capacitation involves the removal of some inhibiting substances or substances from the sperm. The process of capacitation is temperature dependent. In capacitation, secretions of the female reproductive tract are removes cholesterol, glycoprotein, and proteins from the plasma membrane around the head of the sperm. Next one is pH is an important factor in capacitation. So when we talk about this question, we have to understand that when we perform naturally whenever the reproduction process happens, when the sperm is injected into the female genital tract, for a certain hours, the sperms coagulate, okay, and then they go under this capacitation process. In this capacitation process, very important things happen, which is already given here. Now, what will happen is if you do it in your in vitro condition, in your lab conditions, you have to wash it for a specific reason. And they're asking, why is it? So let us look at the first statement. The process of capacitation involves the removal of some inhibiting substances or the substance from the sperm. So this is right. So it removes this coagulating property of the sperms and some of the things that are attached to the sperm, such as glycoproteins, proteins, and cholesterol. So that is why it has to be done. Next one, the process of capacitation is temperature dependent. So this is wrong. It is not temperature dependent. Next one, in capacitation, secretions of the female reproductive tract removes cholesterol, glycoprotein, and proteins from the plasma membrane around the head of the sperm. So this is also right. So this happens during capacitation. That gives mobility and maturity for the sperms by removal of cholesterol, glycoprotein, and the protein which is present in the plasma membrane around the sperm head. Okay, next one. So that's what make them to even go and open up their acrosome cap very easily when they meet the ova. pH is important factor for the process of capacitation. Even this is not correct. Okay, so our option A, our statements A and C are correct. So where is it? A and C? Our option C. So our 129th question, option C, is correct. Okay, next one. Assertion. DNA fingerprinting is applied in paternity testing in case of disputes. Next one. It employs the principles of polymorphism in DNA sequence as polymorphs are inheritable from parent to children. Okay. So our options here are both assertion and reason are true, and the reason is the correct explanation of assertion. Both assertion and reason are true, but the reason is not correct explanation of assertion. Next one is assertion is true, but the reason is false. Next option is assertion is false, but the reason is true. When we look at this question, look at the first statement. DNA fingerprinting, okay, we use it for solving paternity cases. Yes, correct. To understand who is the mother, who is the father, or whose kid it is. Next one. It employs the principle of polymorphism in DNA sequences, and polymorphs are inheritable from parents to children. Okay. See, what happens is when we talk about the application of DNA fingerprinting, so we know that the DNA fingerprinting is very, very important. Okay. Now here we use the DNA polymorphs as our tool to detect any of the any of the similarities that we have. So we have these polymorph sequences, which are repetitive sequences, okay? And these sequences are heritable. So since both the equations, both the statements are right, so we go for both assertion and reason are true, and the reason is the correct explanation for assertion. So Yes, students. So let's look at the next question. Linkage reduces the frequency of. Okay. So when we talk about this question, look at your options: hybrids, all parental types, and then we have heterozygous recessive parents, and then we have homozygous recessive parents. Okay, so these are our options. So one second. All right, students. So when we look at this question, linkage reduces the frequency of what? So now when we see what is a linkage, linkage is physical association of genes present on the loci of a chromosome. Now, if we have the chromosomes and this is the position of the genes on the chromosome, so let us say that we are taking the comparison between two different genes. One gene is very far and one gene is very less in distance with relate to each other. Now, if you look at these genes, now what happens is compared to these two, okay, when you compare these two genes tend to stay together. Why? Because when the recombination happens, there is bulk flow of information. So that now what happens, very closely attached genes or very closely present genes, now what will happen to these? They will get transferred to the next generation. So that means what? The parental characteristics, how, or the parental genes, how they were, the same thing will go to their daughters or the next generation, correct, right? So that is why now what happens, parental characteristics gets preserved. Non-parental characteristics or recombinant characteristics will be very, very less, or it will not be seen at all. So whenever we talk about the linkage reduces one thing very significantly, that is hybrids. So hybrids can also be called as the non-parental variants. So when you don't have non-parental variants, you don't have recombination. When you don't have recombination, linkage is very, very less. So that is why whenever you have more linkage, you have less hybrids. Okay, our option is correct. Next one, choose the odd one. So rabies, influenza, AIDS, amoebiosis, okay, gardiasis, and then trypano, trypanosomiasis, okay, taniasis, ascariasis, and elephantiasis. And then we have cancer, tuberculosis, and tetanus. So we have to choose the odd one. Let us look at the first option. Rabies, which is uh, caused because of a virus. Influenza is also a viral disease, and AIDS is also a viral disease. So all options are correct here. Amoebiosis is Entamoeba histolytica, okay, and then Giardiasis is not actually caused by a protozoan. It is caused by a nematode or a parasite, okay. Now trypanomyosis is also called as trypanosoma, or it is also called as African sleeping sickness. So that is also not because of your what do you tell? It is not caused because of protozoan. Yeah, okay. So next one, Taniasis, Tania solium, it's called as flatworm, which is a parasite. Ascariasis, it is also a parasite, which is a roundworm. Elephantiasis is caused by a filarial worm. So these two belong to one category, and these two belong to, this one belongs to other category. So these two options become wrong. Next one, cancer, tuberculosis, and tetanus. So we have three here. So here, if you look at this, the cancer is completely different to what we can compare with tuberculosis and tetanus, even though these two are microbial diseases, okay, but they are not related to cancer. So this is also becoming an odd one. So we go for option A. Why? Because all of them are viral diseases and they're all grouped properly. Okay, next one. The eyes of octopus and the eyes of cat show different patterns of structure, yet they perform a similar function. This is an example of homologous organs that have evolved to convergent evolution, homologous organs that have evolved due to divergent evolution, and then we have analogous organs that have evolved due to convergent evolution, and then finally, we have analogous organs that have evolved to divergent evolution. So when we look at this option question, eyes of an octopus and cat's eyes are, or the eyes of mammals are an example for analogy, analogous organs. So what are analogous organs? We have, which are different structure, but they perform the same function. Okay. So these are called as analogous organs. So this eye of octopus is dermal because of the dermal modifications, and this is the retinal modifications in mammals. So they're completely different structure, but they do the same function, that is sight or detection of vision. Okay, so these are called as analogous organs. Now here, coming to convergent and divergent. Divergent means you start at one point and then you try different things. For example, you have same structure perform different function. Okay, so that is called as divergence. Then you have different structure but you perform the same function, that is called as converging. Okay, so that is called as convergent evolution. So analogy means convergent evolution. So our option is option C. Okay, next one. Assertion. ELISA test is based on antigen antibody interactions, whereas where a pathogen can be detected by the presence of antibodies, proteins, glycoproteins, etc. on it. Next one, reason. The pathogen antibody to be identified is immobilized on the surface of specially constructed ELISA plates and is then tested. So we have our option. If both assertion and reason are true, and reason is the correct explanation of assertion, and then if both assertion and reason are true, but the reason is the correct explanation of assertion, then we have if assertion is true, but the reason is false. Then finally, we have if both assertion and reason are false. So the question here is ELISA. ELISA is enzyme linked immunosorbate assay. It's an immunological assay. What we do is we target to find antigens using antibodies. Here, what they do is correct. It is antigen antibody interactions where pathogen can be detected by the presence of antibodies. Here it is antigens. So proteins, glycoproteins are all called as antigens. So we try to detect antigens using antibodies. Okay. Next one. So this is wrong. Next, pathogen antigen to be identified is mobilized. Correct. It is mobilized on the surface of specially constructed ELISA plates, and then it is tested. So both places they have used antibodies. It's not antibodies. It is antigens. So that is why if both assertion and reason are false. So option B is correct for this question, 140. So next question, which of the following statements is incorrect? Okay. Hint to always cut DNA molecules at a particular point by recognizing a specific sequence of 4 base pairs. Okay. Besides hint 2, today we know more than 900 restriction enzymes. The name E. coli comes from Escherichia coli RY13. Next one is restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA. Now, when we look at this question, 147, let us look at the first statement. Hint to always cut DNA molecules at a particular point by recognizing a specific sequence of 4 base pairs. Actually, it is 6 base pairs, students. So, okay, I'll write it as 6 base pairs. So, option 1 itself is wrong. Hint 2 recognizes specific sequences, but these sequences are 6 nucleotide sequences, not 4 nucleotide sequence. So, it is wrong. Next one, besides hint 2, today we know more than 900 restriction enzymes. This is true. It is a very textual statement. It is given in your textbook. The name EcoR1 comes from Escherichia coli R1 RY13. Yes. When we talk about EcoR1, which is a restriction enzyme, E stands for Escherichia, CO stands for coli, R stands for the strain. Okay, R1. Okay. So that is how you name a restriction enzyme, and this is also correct. Restriction endonucleases are used in genetic engineering to form recombinant molecules of the DNA. Yes, restriction enzymes are very, very important tools with respect to genetic engineering. Without them, we won't be able to cut at specific sequences to insert our gene of interest. So that is why all three are correct options here. But our option, which is wrong, with incorrect, is option A. Okay, let's go for the next question. A colorblind man marries a woman with normal sight, which has no history of colorblindness in her family. What is the probability of their grandson being colorblind? So let us look at this question. A colorblind man marries a woman. So our options are given in the form of nil, 0.25, 0.5, and then we have is 1. So let's see the question. A colorblind man. So we know that colorblind is sex-linked recessive disorder, and this recessiveness or the character is on the X chromosome. So we'll write it as colorblindness C, and then we have Y. Okay, Y, it does not have colorblindness gene. And this colorblind man, he will marry a woman with normal sight, with no history of colorblindness. When he marries her, Now what will happen to his children? So this is parents, okay? Now I'll write it as children. Now what will happen to these children? This x will go with this x will go with this, again this y will go with this x, and this y will go with this x. So what do we get here? We have xc x, and then we have xc x, and then we have xc, no sorry, we have xy, and then we have xy. So here in our case, in the next generation, what you can find is both the girls are carriers. Students, both the girls are carriers of the color blindness. Understood? Now, see, a colorblind man marries a woman with normal sight with no history of color blindness in her family. What is the probability of their grandson being colorblind? Okay, grandson being colorblind. Now, let us say that either one of them have to marry for their grandson to be colorblind because they only have the defective genes. So now, what is our next step? Our next step is taking one of the girl child and getting her married. So we take this girl child, that is XCX, okay, who is going to marry a normal man. Okay, I'll write it small x, okay, marry a normal man. So what happens between them? So we have XCX, we have XCY, and then we have XX, and then we have XY. So this is the next generation, isn't it? So we have carrier girl, we have affected boy, we have normal girl, and we have normal boy. Out of four, one is effective, correct? So that is 1/4. 1/4 of 100 is how much? 0.25, correct? So 0.25 is the probability of a grandson being colorblind if the situation is like it's very simple question. You just have to apply some logic, students. Okay. So answer is B. Which one of the following statements about morula in human beings is correct? It has almost equal quantity of cytoplasm and has uncleaved zygote, but much more DNA. Okay. Next one. It has far less cytoplasm as well as, okay, as well. It has more than less equal quantity of cytoplasm and DNA. It has more cytoplasm and more DNA than the cleaved zygote. So when we talk about the formation of morula, we know that the zygote goes into a division, and this division is cleavage, and basically the cytoplasmic volume do not increase, the nuclear quantity increases, and only the cytoplasm keeps on dividing into 2, 4, 8, etc. And this is called as morula, where no cytoplasm, extra cytoplasm won't be created. Only the DNA will be more. So it has more, almost equal quantity of cytoplasm as an uncleaved zygote, but more DNA. So this is the option when it comes to the statements with respect to morula. Okay. Next one, we have our match the following. Okay. So let us look at this. We have Nile perch in Lake Victoria. Nile perch in Lake Victoria is a fish which is left in Lake Victoria, and it is an alien species that actually try to destroy the native species. Narrow utilitarian. Now, what is narrow utilitarian? Because we get so much benefits from the nature, we try to preserve them. So this is called as narrow utilitarian. And why do we do this? For obvious reasons. Why don't we cut trees? Because it gives us oxygen and it gives us rainfall. So those are called as obvious reasons. So I'll put it as B. Okay. Next one. Main cause for biodiversity loss. Main cause for biodiversity loss, you know that fragmentation, habitat loss, deforestation. So basically it goes with habitat loss. So I'll write it as C. Next one is hotspots. Hotspots are the one with problem of high endemism. Well, why high endemism? Because the species are restricted to those specific habitats and they do not survive anywhere. So you can see in hotspot there is high amount of endemism. Okay. So now we have our option, that is A is S, okay, P is B, C is Q, and then R is D. So our option B is correct for our 157th question. So let's go for the next question. All right. So our next question is, in a particular plant, two genes control leaf shape and color. Round leaves are and dominant to the jagged leaves, that is small r. Yellow fruits, capital Y, are dominant to the white fruits, which is small y. A true breeding round-leaved, okay, a true breeding round-leaved yellow fruited plant is mated with jagged leaf with white fruited plant. What are the genotypes of the plant involved in the cross? This should be the easiest question ever. Why? Because it's asking about true breeding varieties. So he is given if a true breeding round, true breeding capital RR, and then true breeding yellow is capital YY. If it is crossed with jag leaves, that is smaller, and then you have white, that is small y, small y. So what are the genotype? Capital R, capital R, capital Y, small r, small y, small y. So our option B is correct for 158. Very easy question. All you have to do is look at the question properly. Okay. So our next question. Virus infected cells secrete antiviral glycoproteins called DASH, which move into the nearby non-infected cells and prevent viral replication in them. The correct option filling the blank is interferons, then we have interleukins, then we have complement proteins, and then we have cytokines. So when we talk about a virus infected cells, when a virus infects a cell, the cells under infection, they release some of these signaling factors, which we call it as interferons. So these interferons, they go and bind to these neighboring cells and they create these antiviral proteins, which enable them to prevent any further infection from the viruses. Okay. So these are called as interferons, whereas interleukins are a type of cytokinins. Okay, so cytokinins are again the signaling factors which enable our immune cells to multiply and increase their number. Okay, complement proteins are a series of proteins which are involved in complement pathway in immune system. Okay, so which is not relevant right now. So these three options are wrong and interference are correct when it comes to virus infected cells. Okay. Next one. Which of the following statements is correct with respect to immunity? Okay. Preformed antibodies need to be injected to treat the bite of a viper snake. Next one. The antibodies against smallpox pathogens are produced by T lymphocytes. Okay. Next one. Antibodies are protein molecules which each of which has 4 light chains. Next statement is rejection of a kidney graft is the function of a B lymphocyte. So let us look at this question. We have to find which is the correct statement. So preformed antibodies need to be injected for a person who is bitten by a viper snake. This is correct. Why? Because antivenom is a type of a passive immunity where we extract preformed antibodies already and we inject into these patients. So when we do this, it goes and binds the antigen and it will neutralize the effects of this poison. So passive immunization, another example is the antivenom. So this option is correct. Okay, the statement is already given correct. Let us take a look at other options as well. The antibodies against smallpox pathogens are produced by T lymphocytes. Whichever the disease might be, whichever the pathogen might be, whenever it comes to the production of antibodies, antibodies are produced by B lymphocytes, not C lymphocytes, not T lymphocytes, sorry. Antibodies are protein molecules, each of which has 4 light chains. Antibodies are protein molecules, correct, but they have 4 chains, 2 heavy chains and 2 light chains held by disulfide bridges. It's not just 4 light chains. So this option is wrong. Next one, rejection of a kidney graft is a function of B lymphocyte. Rejection of a kidney graft is a function of T lymphocyte, students, not B lymphocytes. Our T lymphocytes are necessary for differentiating. It will help our body to make it understand the difference between self cell and non-self cell. Self cell means our own body cells, non-self cells means antigens. Understood? So that is done by T lymphocytes. Usually, the donor tissue produces a protein that will be recognized in the receiver body as antigens. That's why the immune reaction takes place. Cancer is genetically caused due to the activation of dash to dash and or inactivation of dash. So cancer is genetically, okay, let's see the options. So we have option A, that is oncogene, tumor suppressor gene, and proto-oncogene. Tumor suppressor gene, oncogene, and proto-oncogene. Next one is proto-oncogenes, oncogenes, tumor suppressor gene. Okay. Next one is oncogene, proto-oncogene, tumor suppressor gene. Okay, cancer is generally caused due to the activation of oncogene. Okay, so activation of proto-oncogene to oncogene. Sorry, cancer is generally caused due to the activation of proto-oncogene to oncogene, and then due to the inactivation of tumor suppressor gene. So what happens is you have proto-oncogenes which have the capacity to change the cellular dynamics. It can change the activities of the cell. Now this, if it is converted into an oncogene, it will start actually what it will do, it will start dividing. Okay, the cell will start dividing. Next one is the inactivation of tumor suppressor gene. So tumor suppressor gene is a gene in our cells which is responsible for controlling the rate of mitosis or to stop mitosis. It's like brakes. So if these brakes fail, now what will happen? Continuous mitosis will result in cancer conditions. So that is why our right option is option C for the question 166. So let's go for the next question. Long statement. Which of the following statements are false? Pyramids of energy and yearly biomass production can never be inverted since it would violate the laws of thermodynamics. The pyramids of standing crops and numbers can be inverted since the amount of organism at any one time does not indicate the amount of energy flossing through the system. Through the certain limitations of ecological such as it does not take into account the same species belonging to two or more trophic levels, saprophytes are not given any place in ecological pyramids, even though they play a vital role in the ecosystem. So, which of the following statements are false? So, if you look at them, okay, first statement, pyramids of energy or yearly biomass production can never be inverted. It would violate the laws of thermodynamics. Yes, you can never invert the uh, you know production levels in the pyramid because we know that energy flow according to first law of thermodynamics is unidirectional. So that is shown here. So that is why our option is correct. Now, when we talk about the standing crop at any trophic level, the amount of biomass left for the next trophic level is called as a standing crop. So the standing crop is a resultant of the flow of energy for a long period of time. So at current time, you cannot tell that how much of energy is flowing through the system. You cannot indicate through using the pyramid. So our option B is also correct. There are certain limitations in ecological pyramids, such as it does not take into account of the species belonging to two or more trophic levels. The best example is when you compare the grazing food chain and the food web. We know that in the food web, when we talk about, we consider decomposers also. Whereas when we talk about grazing food chain, we don't take they take about decomposers. In a food web, you have omnivores which have a complex relationship in an ecosystem. So these things are not considered when it comes to trophic levels, energy distribution. Next one, the same point here, the saprophytes are not given any place in the ecological pyramids, though they play a vital role in the ecosystem. Even these statements are right. All the statements are right, so that we go for none of these. So none of these are wrong. So that is why our option D is correct. So our next question is the bottleneck effect and the founder effect are the two special cases of. So one is genetic mutation, genetic recombination, genetic drift, and then we have natural selection. So the bottleneck effect, that is because of any sudden changes in the environment, the population is suddenly reduced and one group is left out, and they become the new pioneers or the founders. Okay, so this is called as a founder effect. Okay, and the founder effect of the two species or two special cases of what? It is with respect to genetic drift. So in genetic drift, you can see the bottleneck effect because of the sudden depletion of the population. It leads to other scenarios where one of them becomes a founder effect. So that happens because of the genetic drift. All right, students. So here we come to the end of our question paper discussion. All right. So if you have any doubts with respect to any questions, please let me know. Okay, let your teacher know. They will get back to us. I'll see you in the next content video. Thank you very much. Have a nice day.